Hello, everyone, and thank you for attending our first Tech Talk for the year. So we are really honored and very excited today to have Dr. Braun from University of Delaware to give us a talk on mathematical modeling and data science for tear breakup on the ocular surface. But before I introduce our speaker, let me go over some housekeeping issues. As you all know, we continue extending CME credits for our Tech Talks and our ID series presentations. We will distribute the information via email to those that registered for today's event, or you can obtain the information from Deborah Rees at Questionnaire Care, and her email address is already in the chat box. We have a few a few events that are already scheduled. On January 20th, we will have an ID series presentation. Dr. Craig Pollack from Johns Hopkins will talk about housing policy as a tool to improve health equity. And then Anne-Marie Brescia from Nemours, also for an ID series on January 27th, will talk about prognosis in juvenile idiopathic arthritis. And finally, we have another ID series on February 24th from the NIH, who will talk about the All of Us Research Hub, and that's a really great source of data. So you do not want to miss any of these great presentations. So again, we are really honored today to have Dr. Braun. Dr. Braun is Professor of Mathematics in the Department of Mathematical Sciences at University of Delaware, and is a founding director of University of Delaware's Master of Science in Data Science program. He is also affiliate faculty in the Data Science Institute at UD and in the Biomedical Engineering Department. He obtained a Master of Science in Mechanical Engineering from Santa Clara University in California and a PhD in Applied Mathematics from Northwestern University in Illinois. He has received multiple grants from NSF to model tear film structure as well as NIH grants. He has a really long list of peer-reviewed publications as well as books and book chapters. He has been, and he is a teacher, a mentor, and has served on multiple dissertation committees, and has been invited nationally and all over the world to give keynote addresses at plenary sessions and other presentations at prestigious conferences. So we are really honored to have Dr. Braun today and very excited about the presentations. Thank you so much.
Thank you for that very kind introduction, Claudine. And I'm grateful for the opportunity to talk to you today about this work. So this year marks 20 years working on eyes, so it's been some time. And I've worked with a lot of different people from math, from optometry, from this Master of Science in Data Science, and so I'll tell you sort of who they are later. Yeah, and so the outline today is then going to be as follows. I'll give you, it's a little funny talking to medical people about the eye for me because I don't really truly have medical background, but I'll give you a little introduction to the tear film and the ocular surface. And I'll run through some levels of modeling that, and in fact, getting simpler models allowed us to do this automation where we extract data and fit to it and to enable us to generate much more data. And so that I'll give you some example results. And if you have questions, like clarification questions, feel free to ask. But I prefer not to have a debate in the middle of the talk. So sort of short answer questions are no problem at all. So why should we care about the tear film? Well, every time you blink, your eyelid drops and it acts like a paintbrush and paints on a thin liquid layer. And that protects your eye and facilitates clear vision. And if things go wrong with it, it makes it harder to see properly. And when the tear film fails, typically you just blink until it's better. So you just don't think about it, which is good. But some people can't seem to have a good homeostasis where they can see properly. And so one category of this problem would be the so-called dry eye disease. And in dry eye, millions of people have it. It tends to be more prevalent as one gets older. Lots of unpleasant symptoms may occur. There's a range of them depending on how badly you have it. And it definitely can affect negatively your vision and the health of your eye. And so, this is a pretty unpleasant looking picture of how uncomfortable it could be. And so, but there's a lot of things that are unclear about what causes dry eye. And in fact, there's a lot of interest lately about neuropathy and not just stimulus from the tear film, but also a reaction to it within the eye. But you still need to know the stimulus. So one way to view what we're talking about today is, what is the stimulus? How can we quantify what happens on the front of your eye to help provide the basic knowledge needed to evaluate these things? And so what are some of these issues? Well, some diagnostic procedures are not really repeatable enough. And so, I mean, it's just maybe, it's not the only place in the biomedical world this might happen, but as an example, in the Far East, it's popular to use a static image of a pattern on the eye to make diagnosis and to classify dry eye disease. But you can see patterns like this in normals. So here's just a selection of this. I'll explain these images more later. But you can see many of those same images that they use for diagnosis in normal subjects without dry eye. So that's a bit troubling. Some studies don't use control groups. And there's a critical variable that people associate with dry eye disease, which is the osmolarity. So basically the saltiness of your tear film on the front of your eye. If it gets high enough, it's a very negative stimulus in the front of your eye. And but you can't measure it directly in tear breakup. So these images are right where it's very dark. It's thought that the water part of the tear film has left and it's very salty, but you can't measure it. So one aspect of, so many places discuss osmolarity. Here's a sample of a couple. So what we really need is some quantitative and repeatable estimates of what's going on in these things like tear breakup. And that's what we're after. Okay, so what I'm going to do is now talk a little bit more about the tear film, how it gets there, and some of this imaging processes. Okay, so let's start by talking about the tear film. So here's a schematic of looking at the front of the eye, and there's a dashed curve. Can you see my cursor? But there's a dashed sort of circle around the eye, open eye. And that's the fornix, and there's underneath there's some tear film, and then here's the open eye, and there's the iris, the pupil, the cornea is the clear part in front of all this, and the tear film's on the front of the cornea and the conjunctiva. And so the water is supplied from the lacrimal gland, and the majority of the water supplied in your tear film comes from there. You get a little bit from the ocular surface, but it's very minor. Tears accumulate near your lids. They flow toward your nose, and they go down what's called the puncta. And the punctas, there's drain holes, if you like. Canaliculi are tubes that connect to the lacrimal sac in your sinuses. And so when your eyes water, when it's cold out, this extra water is draining down through the canaliculi into the lacrimal sac and coming out of your nose. And the water definitely goes down the puncta. So in this image that just showed up, there's a white arrow in E. There's a white arrow pointing to the puncta. So that's where your eye, when you get near your nose, your eyelid edges kind of bend a little bit. They're typically right there. And the orange areas in this image are quantum dots, and they're lipophilic. So they like to go where lipids are. And they don't like to be in the water part of the tear film, and they accumulate on the edge of the eye and on the lashes. And in the uppermost white arrow on the right in F, they're accumulating on lashes. So it's kind of interesting. Lipids go onto your skin and lashes, water goes down these puncta or evaporates to the world. And the menisci are these sort of rivers of tears, if you like, that flow toward the along the edges of your lids. And so when you blink, that upper lid drops and paints on tear film on the rest of your eye, if you like. So here's a view in the sagittal plane or from the side that or cross section, if you like. And so in a blink, the upper lid drops. It drops quickly, and it rises about half as fast, and it typically takes around more than a quarter second, a little bit, to do a blink. So it's very fast. And you paint on this thin layer, and you have to establish a sort of complicated structure, and I'll talk about that in a minute. But what we're interested in this talk is what happens away from the lids. So we've done theories for what happens during a blink and over the whole eye, but what we're really interested in the whole open eye surface. And so what we're interested in are local areas away from the lid edges. Okay, so that's where we're going to develop theories and try to get data. Okay, so here's zooming in into the middle of the tear film. There's sort of a cartoon here that imagine air being up here and near the top of the slide. The eye is itself is down here at the lower part of the slide. And there's some epithelial cells sketched here, and then there's various structures here. So if working backward from the air, there's a lipid layer, a floating lipid layer that's a barrier to evaporation, and it can also drive flow along the eye surface. And so, and so we call that tangential flow. I'm going to simplify what the lipid layer does greatly in all the work today, so we really won't treat it directly. The aqueous layer is so the lipid layer is only around tens of nanometers thick. The aqueous layer, the light blue area, is about a few microns thick, so it is much thicker. Think of it as 100 times thicker. And there's proteins and other big molecules floating around in there. And deforming that upper interface with the air, that is the lipid layer, you can cause flow due to capillarity, which is it's the same effect when you look at a test tube and the water goes up the edges. It's precisely the same effect. So you also get evaporation of water to the world through the lipid layer, and that when you lose water to the world, you leave other ions and other molecules behind that increase the osmolarity inside this layer. And you can also instill sodium fluorescein to visualize the dynamics, and I'll come back to that quite a bit. You can also get a little bit of osmosis from the ocular surface into the tear film when the osmolarity rises enough. And that goes through an area called the glycocalyx, which is, think of it as like a velvet of big mucin molecules that protects the ocular surface. So this thing gets established very quickly, in tenths of a second, every time you blink. And we're taking it as a given, effectively. Okay, so, all right. And so what I'd like to do now is we want to image this. The eye is so sensitive, the only way we're going to get data is to image it. We cannot sort of poke around in there. Everyone's going to flinch if I try to start doing things to the front of your eye. So we have to visualize it. So your data is going to consist of movies or photos of what's happened on the ocular surface. In the case I want to talk about today, you instill sodium fluorescein. Under physiological conditions, the 2 sodium ions come off. And a typical installation might be 2% solution, or you use a fluorescein strip. Although how you get these days seems to be a bit complicated, but I'm not going to go into any of that. So this image is from Indiana University School of Optometry. We collaborate a lot with them and get a lot of data from them. And so what you do is you put this sodium fluorescein in, and then you shine a blue light on it, and it glows green. And so the green arrows are meant to show that fluorescence. And I'm sure many of you are familiar with other uses of fluorescein or similar molecules in a lot of biological applications, but this is just in solution. Typically, you filter out the blue light source because it's brighter and sort of pollutes the image, so you really want to see as much of the green fluorescence as possible. Okay, so There are some other technical details about how they do this procedure, which I'm just going to leave out because it just sort of complicates the picture. You can also use fluorescein, of course, to reveal vital staining, and you can do it like other stains. Okay, now for us, staining is a complication. We prefer not to have any. So, okay, and so what is the data? The data is they have a procedure to instill the fluorescein in subjects, all approved by IU, their IRB board, and all that stuff. And so they picked healthy people who don't wear any contact lenses, and they only looked at the left eye, and they watch what happens in this when they visualize this tear film this way. If they see areas get dark slowly, so darkness is meant to indicate that the tear film has thinned and maybe broken up. And so maybe there's no water left when it's really dark. And if that happens slowly, it's typically evaporative. So water is being lost to the world by evaporation. If it's really fast, then it might be something else. And in this movie I'm going to show you, you're going to see that there's some slow development, but then at the very end, you'll see something happen very fast. So, so what this is suggesting is that even in the same subject, or even in the same trial, the trial would be a movie, then you can have different mechanisms active. Okay, and so there's the movie's running now, so there's a blink, a dim lighting, and then the lighting goes up to something brighter. And you can see this dark area develop here, and we think that's breakup. Now, that's great. However, I want to show it again. So if we watch at the end, you'll see something really quick. But when exactly did it get dark enough to TBU to happen? In the clinic, it's all by the optometrist watch, or the ophthalmologist watches and decides when it happened. It's not a quantitative measurement. So, sorry, this did not cooperate. Oh, maybe it's trimmed out. Sorry, I'm not sure why at the very end we're not seeing it, but there's a really fast burst at the end of the original movie. I'm not sure if we trimmed it out here. But anyway, I'll come back to that later. So, okay, and so what we need to do is to add here. Okay, so I need models for these dark areas, how they develop. And I'm going to go through a sequence of them. And we'll start out with a sort of a conceptual, first of all, we need a concept to try to build a mathematical model from. And I think there's been conceptual models around for quite a while about how some of these mechanisms work. And so what we're really doing here that's new for the most part is quantifying these things. And so let's convert this sort of slow development first into a sort of conceptual model. So we're looking at the sort of a cross-section of the tear film. Air's up here, the eye's down here. There's a tear film in here. The lipid layer has been simplified to something that just does not move along itself. So we've just simplified that more or less out of the picture directly. Indirectly, we put in some evaporation. And so, and there could be osmosis, there could be some flow. So what are these critical variables we're going to build the model on? Well, the thickness of the aqueous part of the tear film is H. We're really interested in the osmolarity. So we're going to use these math models to be like an instrument that's going to estimate that osmolarity, this critical variable in development of dry eye. We're going to also calculate the fluorescein concentration so we can try to generate our own theoretical intensity to match to the experiment. Okay, and then we'll also need to compute that intensity. So those are our key variables. And so for the evaporation, we're going to fake the lipid layer by assuming we have a localized increased evaporation and less evaporation outside of that. And I didn't show it today, but I have direct evidence of simultaneous imaging of lipid layer and aqueous layer that shows that situation without a doubt. So it's not, it's a conceptual simplification just to make the math easier, basically. Okay, so so that evaporation drives thinning, but it also, so that locally we lose more water here than over here. That makes if you take away water here, there's more salt ions, more fluorescein ions, so concentration goes up. But capillarity makes water and tear film flow toward this thin region. So it's kind of an opposing force, if you want. Evaporation thins the tear film, flow makes it want, in this scenario, opposes that thinning. It brings in more solutes, and that process of bringing it in is called advection. So basically solutes can move with the flow, but they can, if they build up enough, they can also diffuse outward by Fickian diffusion. So you can see that already the conceptual thing's building up a bunch of effects. This is why tear breakup is so complicated, because there's all these processes going on. So we're trying to decide which one of all these effects is the most important, essentially, in a given instance that you measure. Okay, and so we have equations. We can calculate the thickness, the osmolarity, and the fluorescein. We can take those results and calculate intensity and match it to experiment as best we can, and then infer what we want to know. That is our game plan. Okay, so I need to fill out the picture just a little bit more. So I need to tell you about this fluorescent intensity. So if I were to, it depends on the thickness and it depends on the fluorescein concentration. And it's a little bit complicated how it does it. So we solve differential equations, either partial or ordinary, and then plug in the results to get the intensity. So we solve for F and H, which I've already mentioned. These things are constants, and I get out an intensity. So I use this result from simulations and force that to be as close as possible to what we measure in experiments. Okay, now I have not shown you the messy equations to get the thickness and the solutes and so forth. If someone's really interested, I can show you later, but there's some slides at the end that do. Now there's, so everything I've described so far is what happens when things are slow. And this breakup or failure of the tear film is driven by loss of water to the world via evaporation. So now I want to put in a second possibility. And so we've done a couple of papers about this where the lipid layer is now back in the picture. And so what we care about is the tear film thickness. We care about a concentration of surface active lipids. So these are like dish soap, if you want. If you put dish soap in the middle of your sink in clean water, it spreads out really fast. It does exactly the same effect. But in this case, tear film lipids are responsible for this. And so we can build a model where there's locally more lipids, and they make flow go outward, like the dish soap would do, and that makes the tear film thinner. And that effect is called Marangoni flow. And so that makes the flow go the opposite direction. So in the last scenario, flow went inward to try to slow thinning. Now the flow goes outward and drives thinning. And so we can calculate from the models what we need to know given this conceptual scenario. And and so the idealizations here are somehow that this buildup of extra lipid that you want to smooth drives this flow, and that the flow, there's an assumption that this so-called glob of a lot of lipid remains there for a while. And so empirically, this kind of high or extra lipid is seen. There are definitely cases seen experimentally. If you want to see one at the end, I could show you that. And so we build off those observations to have these conceptual things that we then implement as math. Okay, so and it's also possible that these two things are present in combination. The eye doesn't know it's supposed to do these things separately. So we can think of them as limiting cases. There are examples that are completely like the first case where it's evaporation. There are some really fast cases that are all like flow. But there's a bunch in the middle that are some combination. And so the real strength of the math models is you can tune those and figure out when you're at the extremes or in the middle somewhere. Okay, so that's our sort of conceptual model picture. And so what are the symptoms, if you want, or the empirical things you might see? Well, on the left, we have these list of things that are typical of evaporative scenario. Slow thinning, solute concentrations rise a lot. We'll see that later. And something that's not very easy to control is that higher initial fluorescein concentrations or installations are better for this case. When things are fast and the lipid layer is driving this outward flow from some middle region, the solute concentrations don't change nearly as much typically. And a lower concentration is better for visualizing that. So it's kind of a contradictory situation that you just do the best you can. So generally, we start with a fairly high concentration and live with what happens. And so I'll start off with more complicated models and then simplify and do the automation. Okay, so, all right. And so this, now we believe it or not, they're messy equations, but we think of them as fairly simple for fluid dynamics. And so we still have to solve them with a computer. They're complicated enough, we have to do that. And so we'll test how well we can do against observed dynamics, meaning intensity data we extract out of these movies. And from that, we're going to classify what mechanisms drove these tear breakup instances. Okay, so, all right. So here's the conceptual picture. So you have a movie. You have some place, maybe this little circle in the square box here, and this is inset is a zoom on that, and we could sample intensity along this line and compare it to theory. So in order to do all that, we got to we have to get this region selected and then extract data. We also need to estimate what actually was in the tear film, how much fluorescein was in there, and a tear film thickness. So my graduate student, Rayanne Luke, who's now finished, developed an initial thickness estimate method. And at Indiana U, they developed a method for the initial fluorescein concentration. So that was available prior to our starting, but Rayanne had to develop this initial thickness or he would have been nowhere. So with that information, you can extract, you can extract intensity in any case. And then here's what that intensity might look like, so that it develops as time goes on. And so along that line, the data might look like that. So, okay. Now what we do is we have a mathematical model that we force to be as close as we can to those experimental curves. So the theoretically calculated values, they're more perfect looking, they're perfectly symmetric. These are not, but they're close. And so we make this match this as close as possible. And that's what this pink box here is saying, is that we have a mathematical algorithm and code that was already available in MATLAB to do this task. From that, we get the peak evaporation in the middle. We get a background evaporation out here on the sides where it's slower. And this is essentially fitting a Gaussian. And we also have the width of the Gaussian, which we think of as like the radius of a hole, of this hole, if you like. So we determined those last 3 boxes from doing this fitting to the experimental data. None of those things, well, the evaporation rates can't be determined empirically in these local regions. So there, to my knowledge, there is no other way to get that information. So here's a plot of the experimental data together with the optimized solution. So those dashed curves are the calculated answer, and the solid sort of stepwise curves are the intensity data. And so you can see in this case, it's really a pretty good match. And we do this over about 17 seconds. So it's a fairly slow process. And so, yeah, and so that's a good case. And we were able to do that, to make this match by finding those 3 parameters. Now, one thing we really want to know is the thickness. And we can't measure that directly. All we have is this intensity. So another thing with this method that we can generate that's not available from the method is this thickness, the tear film. So it drops from about 3.7 or so microns to about 2 microns in the middle, according to the theory. And so that's new information we couldn't get directly from the experiments. And yeah, and it is not the same thing as the intensity, even though the curves look similar. And so that's our first thing that we get out of the math that we couldn't get from the experiment. You can also, and so you can also, Rayanne Luke worked heroically and got 20 PDE fits from using these partial differential equation models. In order to get that, we had to search for these really idealized strip shapes, or the circle shapes like you saw in the box when I explained the fitting procedure. So we had to find these idealized instances and then fit these messy math models to them. And she did a great job of that. And so the results we get compared really well with experiments, which are in black. And her thinning rates, depending on the kind of model, are either blue or yellow. And so the thinning rates are fastest when the flow matters. So the M is the case where the lipid layer drives outward flow. And so those were the fastest. And then the evaporation is a bit slower, but you can see that the blue and yellow are really work pretty well with measured experimental data from a paper by Nichols and company and Ewan Kingsmith in 2005. So sorry I didn't give that reference here. So okay, and so we only had 12 evaporative fits and 8 mixed mechanism fits, which is both this evaporative force plus this Marangoni flow or outward lipid-driven flow. There are 30 experimental measurements in black. Now, if you're thinking about medical studies, that is not a big sample by anybody's measure. I'll get back to that in a minute. So now another thing is critical variable about how salty the tear film gets, that is the osmolarity. We could compute that now, which could not be measured in TBU breakup. To my knowledge, it's still not been measured directly. And so the mixed mechanism are these yellow circles over here, and the evaporative ones are these blue squares. And these are at the end of the simulation period. Okay, and so you can see that the evaporative cases are more, so the ideal osmolarity is roughly plasma concentration, and we've taken it to be 300 milliosmolar. And you can see that the evaporative cases all more than doubled that saltiness. A subject will start turning a pain knob in response to an osmolarity of about 450. So this is clearly irritating to the subject if all their nerves are functioning properly. The mixed mechanism cases are all over the place. So these short cases may have high osmolarity or not. And that's something that's really hard to tell just by looking at how fast something happened. So, so that's something that's coming out of computing mathematical solutions. So this is a crucial variable, and it's what we can see is that it tends to rise, but it doesn't rise indefinitely. So it seems to rise up to about typically around 700 milliosmolar, occasionally higher. We'll get a lot more information about that soon. Okay, so this is about as far as we could get with doing these partial differential equation models that describe the shape of the interface of the tear film with the air and the distribution of solutes along the ocular surface. And yeah, and so we in order to get bigger samples, we need to simplify the mathematical modeling. And so let me tell you a little bit about that process. So if I've selected a TBU area, so in this case it's a dark circle, and so we want to extract data, if you like, from this bright spot in the middle of it. Okay, so now I'm not, before we were sampling along a line across this circular area. Now we're saying forget that, I'm just going to take it from the middle. Okay, so, and what we're going to do now is fit a math model. We're still going to be able to get evaporation rate, but we have some simplified flow parameters, and we're not going to vary the permeability of the ocular surface. So we have just a few parameters now. Whoops, sorry. And so I have this, what I would call a simple equation that has some elements in it. And so the elements are evaporation. So this thing is the rate of change of the thickness of the tear film, meant to be, sorry, this thing here is the rate of thinning of the tear film. And it's meant to be the rate of change of that thickness right there in that dot. Now it's affected by evaporation. So if you're losing water to the world, then this thing says that this is decreasing. If the osmolarity goes up, osmosis says you put more water in, and so that would have the opposite effect. And if you have flow, this says the A being positive says flow is outward. This is exactly like pulling taffy. So if you imagine this, you've been to the beach probably, and you've seen taffy pull, that when you pull on it, it gets thinner. That's exactly what this term says. So we pull taffy, we let water evaporate, or we supply water when it gets salty. And that's it. That's all that's in the model. And we can make it fancier by making the flow more complicated. So in this case, the sketch that goes with that, so here's a picture of the flow gets faster as you get away from the middle. Now it does the same thing, but it gets slower as time goes on. So that's just, you pull taffy slower and slower as time goes on. So that's what this sketch represents and what this equation represents. So in the computer, we have the great flexibility that we can say, well, I don't want flow, I'm going to make that 0. Or I don't want osmosis, I'm going to make that 0. Or I don't want evaporation, I can make any one of these things 0. Or make the model as complicated as I can. So we use that to now try to fit different models to extracted data from here to determine that mechanism. So yeah, so that is our strategy now. I have these models, believe it or not, are much simpler mathematically, and I can solve them very fast, and I can compare very quickly to data from the center of this spot. Okay, so how does that go? So if I hand pick my locations again, I have a bunch of options where I may have only V is non-zero, the evaporation, or only V and PC are non-zero, evaporation and osmosis. And in turn, I can increase the complication of the model. And these curves are the results of the different models, and the blue curve is the intensity from the middle of the spot. You can see I can make them pretty close. And so bear with this table briefly. I'll try to walk you through what's in here. Evaporation only, I only have V, and this number in the last column is a measurement of how close am I between that case, which is the orange dash curve, and the blue curve. So I can measure create a number that measures the separation between them. And so I want to make that as small as I can. The evaporation only wins in this particular example, but the more complicated flow case is very, very close. And so, so this is taffy pulling that gets slower as time goes on. And but it also has fast evaporation. So it really has both things going on. And one thing I want to focus in on is that this thing looked the best. These two boxes with flow, both models say that flow goes inward. So I've got significant evaporation, I got flow going inward, and that's consistent with this case one that is evaporation driven. So these ODE models are agreeing with the partial differential equations model, the one that's more complicated, and they're all consistent. So this is a really good case that says that these ODE models will produce this scenario in agreement with all the signals. So that's a good thing. Okay, now I'm going to turn to a different case. So this is another hand-picked TBU instance from the data. And in this case, the time interval is much shorter. So here it's just a couple of seconds. And so it's much faster. So you might think there's going to be a different result. Again, we can extract experimental data as this darkest blue curve, which is here, right here. And so we can tune the parameters to get different models that emphasize different effects, compare them to the experimental data again, and what do we see? Well, here's the evaporation only. We get this absurd number that's ridiculously high. No one believes this number. Same thing here. So you still have to use a little bit of judgment sometimes, but this is clearly, in our minds, a fail. When you start to allow flow, either a constant amount or a decaying amount, things get much more reasonable. And so this looks like, again, a mixture of evaporation and flow, but very, very significant flow. So we would call these very high flow rates. And so the winner in terms of the curve that's closest to the extracted data is this time-dependent or time-decaying flow. And again, if I look at the flow, even if I use a dumber flow model or the fanciest one, they both say that the flow is outward. So this is strong, fast taffy pulling. And so that the flow is very, very important in this instance. And so that, again, is what should happen and what the more complicated math models say too. So everything hangs together. And because of that, that allows us to now use these to try to ramp up how much data we can generate and parse for what's happening on the eye. Okay, so, all right. So, again, so we are looking for these dark areas. I'm not gonna show the movie because I'm afraid I'm gonna start running on too long. So yeah, again, we can sample these spots, but now we want to do this. So all told, Dr. Luke went through 42 instances with ordinary differential equations or PDEs or both, which was a heroic effort, but it's not nearly enough to get a good sample of a population. So now we're looking at how much can we get out of 500 trials, 500 movies of subjects from Indiana U. And so we got to get automatic. That's the upshot here. And so we want to try to use some deep learning to find the TBU instances, and then we can use other code to extract data from that and then do these fits. And because it's a simple model and fast, we can do that for a lot of cases. So the basic idea comes from a paper by Su et al. In a 2018 paper. And in a nutshell, what they do is they train a convolutional neural net, and they divide up an image of the eye into a bunch of squares, and they search for where they get a match from a trained neural network and what's seen in the image. So they have to, essentially they have to scan the images by translating boxes all over the surface of the eye and finding the highest probability area where breakup happens. Once they've done that, then they decide, okay, I'm going to trace this box through time. And they find when was the first time they had something labeled as breakup. And that completes what they were interested in. They wanted to mimic what the clinician does when they find out when's the first time that I have breakup. And they wanted to do that as closely as they could to a clinician just watching the process. So there is no thresholding. They're not looking at, does it continue to get darker? It's just, how can we make this match a clinician? Okay, and they did a very good job of that, I have to say. We want more. We want to take that kind of knowledge from this region of interest in this box, and we want to go on throughout time and extract data from it. We want to be sure we get all the way to when we're as dark as we can be. And so we're trying to make that more quantitative and more precise. And so we will then extract data from that region, and we'll plot that central orange, this curve down here might be the center. We will find that central or first TBU region data and fit a model to it and get mechanism. So we're going well beyond what they wanted out of their system. Okay, and so as I've showed you before, that's the kind of output we can get where we try to match that experimental data. Okay, so I think I have to speed up a little bit because we're going to get short of time. So the basic idea is we created our 50,000 images that we labeled manually from this data set. We took 80% of them and trained our CNN. We used a threshold, an objective threshold for determining when we had TBU, not just looking by eye. We validated the network on 20%, the remaining 20% of the images. I'll show you the numbers in a minute. We then used the trained CNN to find TBU instances and then extracted intensity data. And then we fit these ODE models to them. We used a kind of a language called Julia, which has a really super ODE solving package in it that allowed us to do a lot of things that we needed. We then take the parameters from those fits, we collect them to determine the mechanism from each instance. And we're going to be able to do hundreds of instances now of TBU. And then we can analyze the resulting collection of parameters on those instances. So you can, you probably recognize various instances I already showed you, mostly from the right. And so we had to develop our own version of what's on the left, really, to enable all this. Okay, and so what we had to label was, in these images in the eye, we could have lashes, we could have lids, we could have areas over the cornea that were not TBU, or that were TBU, or you could have the sclera, that is the white of the eye. And so there's a confusion matrix here on the right. The numbers are really the biggest by a long way on the diagonal, which says we're doing a good job. If you look at precision and recall for various kinds of images, we did a good job. And so the accuracy we assessed at about 92%. So not perfect, but certainly good enough to generate a lot of instances. So what did things look like? So these boxes, colored boxes on this image over here, are identified regions where we were looking for TBU from the CNN. We needed to look more carefully within these boxes, and in order to extract the data we could fit. And very quickly, we would follow this tile, if you like, through time. Within the box, we scanned for the first time we got TBU in smaller boxes, 16 by 16, and we averaged over those and found where we got the first intensity to hit TBU. So we really had to work at getting a reasonable algorithm to extract a region out of there, central data. Okay, and so anyway, a representative trace is shown here on the right, down here. Okay, so I'm going to skim through this fitting. We actually had 4 models. We eliminated the most complicated one because we had too many parameters, and this is one of the things that Julia helped us find. You can see that we can do a good job fitting data, right. Okay, and so the models with flow win. In fact, in Julia, they win essentially every time. And but they might be very, very small flow, which is fine. Now the thickness drops here. The model we ended up keeping is the purple curve, which is a so-called D model. That was the one the most complicated one with decaying flow. And you'll notice here that in this particular example, the intensity drops a fair amount. The thickness drops to about half, and the osmolarity rose by about 2/3. So it's about 60-something% higher than it was initially. Enough to feel discomfort. Okay, so that's just a typical example. Now I want to go farther and I want to say, okay, now if I do fits for hundreds of examples, what happens? So here's some results for all of the fits we could do. So we could get 467 usable fits. And these were used out of 15 subjects of the 25. Now we have histograms here of the initial thickness estimated, which ranged from around 2 microns up to almost 10. So we had quite a range. The initial fluorescein concentration was peaked around a little over 0.2. That's a very important concentration. It's where we expected it to be. There are a few cases that seem to be not that well fit, but are estimated. But overall, we're doing quite a good job. The fit intervals are definitely stacked toward the shorter end. And if we do a scatter plot of initial thickness versus initial fluorescein, it's really looks like a normal distribution except for this one bit here. And we're still looking into why that is. But it told us right away we need to be careful about our data. So we have a pretty large sample for healthy eyes now. And so some of these distributions are very, very satisfying, but I've already pointed out a couple things that we probably ought to look at a bit more. If we then look at the parameters we computed from the math models, these are very reasonable. So the evaporation does extend up to a very high number, but those are relatively rare in these healthy eyes. And many of them are clustered down here now, maybe around 1 or 2 microns per minute. And that's totally expected. The flow is also quite reasonably distributed, so we're quite happy about that. Now, if we look at the thinning rate compared to what Rayanne Luke did in her thesis, then the orange bars are for about 42 cases, and the blue bars are over 467 cases. So these are probabilities you're looking at. And so the current ones are the blue and the previous are orange. And and so so there's a bit of emphasis at the higher end, probably because we were hand picking, essentially. Now, so there's a difference between the thinning rate. This is sort of a subtle point about the math models, that the evaporation rate is not the same as how fast the thickness drops. And you can see that the thickness drop is definitely a bit higher. That's the DE-CTR is larger in general. And that's because flow contributes. And so that's expected. that what you actually observe should be worse than the evaporation rate you compute. Okay, and so I think that all looks good. If we compare what happens with the end thickness, HE, compared to the initial estimate, the distribution is obviously much higher, and things get much more weighted down to 2 microns or less, which is also very satisfying. And an important new, these are, sorry, I guess I don't want to go there yet. So if we look at a distribution of the final osmolarity, so this is this critical variable of how salty things get. You can see that for these normal subjects, nearly half are way down here, or more than half actually, they don't really feel anything. So nothing real bad is happening. That's also good. That should happen in normals. But there's also this big tail that goes out to here up to 3 times normal or isotonic values of osmolarity, the normal value. So that's like seawater. So you can see why that might get kind of irritating on your eye. I should have mentioned that so why would you ever see this at all for normals? Well, in this trials, the subject was asked to keep their eyes open as long as possible. So even if they were uncomfortable, they were supposed to keep their eyes open. And so that's why you might even see those at all. So, okay, so we've tried these histograms and scatter plots. We did some clustering, which I'm not going to report about today. We also looked at single subjects. And we tried various sanity checks, and there are other things we should check for. But a lot of things are looking very good. Let's look at what happens for individual subjects now. So here's 4 different subjects, and what's on the axes, which I regret I did not put on, that's a rookie mistake. So on the horizontal axis of all of these is the thinning rate or evaporation rate V. So V on all these things could go up to 40 microns per minute. And on the vertical axis is B1, that is the flow parameter. So that's the constant part of how fast you're pulling taffy. Okay, so if the B1 is big, you're pulling fast. If B is 0, you're not pulling at all. And so if it's negative, it means that you have evaporation and flows going inward. And the OD is capturing that. So you can see that the distributions are different. In this case, subject 28, who generally had very long trials, has a really good tear film. Not much evaporation, not very many cases with any flow. Really, everything's packed in by the origin. They have a really good tear film. But then you can see as you sort of go farther, if you go to these other cases, then things get much more scattered. And subject 1 has quite a few more instances where both the evaporation and the flow up here, both evaporation and flow can be sizable. And so we've gone from everything crammed in near the origin here for a really good tear film to something that's not as good. So there's a possibility of distinguishing between subjects, which is a great thing, because that means that, and qualitatively, this doesn't surprise anybody. Different subjects will have a different distribution of what happens on the front of their eye. It's not a surprise qualitatively, but you need a method to tell you that, what it actually is. So we're pretty excited that, at least in some cases, you can distinguish subjects based on what you use the computer to measure on their eye. Okay, so I wanted to give you one last thing about the results. So if we look at all the subjects and look at what happens with the evaporation rate versus this final osmolarity, 300 is the ideal value. As the evaporation increases, so does the final osmolarity. You saw results with a few numbers. That is holding up over lots of numbers. Also, there seems to be, the numbers just don't get too high. They don't keep rising indefinitely. So they are kind of limited like they were before. So those preliminary results held up. And this is, yeah, okay. So that's a really good thing. Another thing is if the flow is really strong out here on the right, then the osmolarity stays lower. You don't get any really high values out here to the right. And so that's also expected. Now, when flow is slower or inward, then the osmolarity can be quite a bit higher. And that holds up over all the subjects so far as well. So, right, and so no low osmolarities at high evaporation. High flow has no high osmolarities. And so they're sort of opposite that way. Right, and so to summarize, we started out looking at really complex math models. The data was good, but it was really limited. And that, so that was characterized by lots of detailed information in the area around TBU. But then we simplified the models to pump up how many places we could extract data and sample. And so that was using ODE models. And so that gave us a lot more insight into the osmolarity and other variables for the whole population. And so we could distinguish some subjects. And yeah, and so that what we really need to do now is take this and use this on dry eye subjects and see, are their osmolarity values worse? Are their distributions of their causes different? Can we distinguish them the way we could distinguish some of these normal subjects? And we're pretty optimistic about that, but we need data. So the ball's in IU's court at the moment there. All right, and so and if this is going to extend into other labs, we have to make it very robust too, and we're not quite there. So I should recognize we had a lot of people contribute to this. So the old timers and the IU people are on the right, and we had many students help. The 4 whose pictures here are all associated with the MSDS program, and at least one more, Juliana Dorsch, is as well. And so they were essential to being able to do this work, and not just the math PhD students. So yeah, so thank you very much for your attention, and if you have any questions, I'll be happy to answer.
Thank you so much, Rich. I mean, this was just amazing and an incredible application of applied mathematics, which I'm really discovering. Any questions? Well, I have a few, so I can
Okay.
start.
Yeah.
So just to make sure I understood, when in your first experiments, you talked about 42 instances. That were 42 different subjects?
No, not 42 subjects, 42 instances. So it was probably came out of about 8 or 10 subjects. So yeah, so those were very hand-picked sort of proof of concept cases. Yeah.
Okay. And so are the subjects doing, or the little movies, are they repeated multiple times for each subject, or is it one movie per subject?
Yeah, I did not give you very much information about the protocol for doing the experiment. So in this set of experiments, the subject visited twice, and each time there was 10 trials done. So 10 movies were done for each visit, and there was, I think, about 28 original subjects, and they threw out 3, so it was really sort of 25 subjects available total. So that's where the 500 comes from, is that. And so when I showed, sorry, let me, oops. So if I go back here, yeah, so in this case, this was one trial that had been processed, and these boxes were identified. And so some of the cases, some subjects might have 5 cases sampled. So these 467 instances are over 15 subjects. And so Yeah, so it's not a real scientific sample in the sense of uniformly done over a population at random. So yeah.
Any other questions from the audience? I still have questions, so I can. No one is raising hands. So, I mean, this is really, again, this is fascinating. Did you think, or would you consider including external factors, or maybe more intrinsic factors, specific to the people? So, for example, age. I'm sure that the breakup of the tear, also varies according to age. So when you saw the individual results, I mean, should it also, could you, do you think it would be feasible or important or interesting to include, other factors such as age, or even, environmental factors such as, dry air, or,
Yeah.
these type of things,
Yeah.
which I guess you can directly figure out from evaporation, but I mean, that's also the type of things that would be important in the tear breakup.
Yeah. Well, as an example, with the room conditions, we certainly know of, and in other models, we've actually used models that depend on the relative humidity and the temperature, and they set an evaporation rate. And you can fit those models to measured tear film thinning rates based on the lipid layer thickness at some um, um, at a fixed relative humidity. So you could calibrate a model that would take all that into account. But and right, and so what we're after is sort of the physical and chemical mechanisms driving each of these instances. So and so we don't, if we measured more, we could make the model more complicated to do more and match the subject better. So if we knew, for example, about the subject's lipid layer thickness, then we could build that into the models. But we don't have that. All we have is an image of the aqueous layer from this fluorescein. Now I'm constantly begging for simultaneous imaging, where you image both the lipid layer and the aqueous at the same time. So that we could get much fancier and get closer to what you're saying. Yeah.
Yeah, but I still, maybe doing the same type of analysis at different ages, because the model will
Oh,
not
I see.
fit
Yeah.
at every age.
Yeah, I see. So we could certainly have cohorts where we extract data and then
Right.
compare.
You don't think, I mean,
It's
that's
another
interesting.
level of model to go to an individual subject. I mean,
I
from a
see.
translational point of view, I totally see where you're coming from. You might want to get an model for an individual that really predicts in detail what's going on there. But I think we're not quite there yet. I think we need a bigger picture about. I mean, there is no other data that I'm aware of that says anything about mechanism like this,
I see.
and certainly not at this scale.
Yeah,
So
no, that's fascinating.
in terms of
Totally
sort of
agree.
quantifying it. So I mean, there's a lot of hypothesizing, I would say, in literature, and it's very, very sensible. But the sort of quantitative measurement to back it up is much rarer. So yeah.
That's really fascinating. Any other questions? All right. Well, thank you so much. This was,
You're welcome.
as I mentioned, really fascinating. And Happy New Year, and thank you, everyone.
Yeah. Thank you for coming.